So let's get started. Hi and welcome everyone to the last seminar of fall. It's a great pleasure for me to introduce Dr. Agatha Lenartowicz as our speaker today. Agatha, as most of you know, is an associate professor of psychiatry and biobehavioral sciences here at UCLA. And I missed that she's also an associate director for the Stanley One Mind Center for Cognitive Neuroscience, CCM. Great. Agatha got her MA and PhD from Princeton in psychology and neuroscience, PhD under the direction of Jonathan Cohen. And then she completed her postdoc here at UCLA before getting recruited as a faculty. So she is a recipient of the NARSAD Young Investigator Award and also a Klingenstein Third Generation Fellow. So this fellowship is awarded to outstanding researchers and junior faculty who are investigating the causes and methods for prevention and treatment of ADHD and or depression in children and adolescents. So her lab here is focused on developing neuroimaging methods to study the mechanisms of attentional dysfunction and apply to ADHD. So she, along with her collaborators, was instrumental in identifying a new attentional control pathway in ADHD with alpha wave brain activity from EEG. So this led to a funded R01 research project, and she's actually a PI on several R01 research awards, which aim to look at concurrent EEG and fMRI and even mobile EEG to study the mechanisms of oscillatory brain activity. I guess again applied to attention control. So today she's going to talk to us about multimodal methods and how to acquire, analyze and interpret them in the context of attention data sets.
Thank you.
So let's welcome Agatha.
Thank you. Thanks so much. That's my talk. There we go. All done. Thank you. Thank you for being here. Thank you for those of you who are here in person. It's nice to have some faces to talk to as well as the Zoom world. So here we go. All right. So the problem with attention / deficits. So let me give a little intro to why we're studying what we've been studying. We all know what attention is. This is an old quote. We actually do. It's whatever's occupying the mind, occupying the resources of your brain. And we know a lot about attention control. So this is what we usually think, I think, when we talk about attention, is the control of attention, which we know includes, there's a pointer here too. Yes, okay. Which includes a behavioral goal, right? Something we want to be doing, and implicitly invokes the idea of competition, that there's something else you might want to do. Otherwise, we don't need any control. You do whatever your stimulus response mappings are guiding you. We also know quite a bit about attention control systems. We have two here, and it's, this is a 2002 paper now. So we've known this for a long time, more than 20 years, that we have at least two important systems. In this case, there are others, dorsal attention, ventral attention, which work together to provide representation of some elements of these behavioral goals or stimulus response sequences, and help bias activity in other parts of the brain that participate in responding to external, internal signals so that you can select the right response for the moment. So we actually do know a lot. That's sort of my point. We know about the systems. Now, we also know that if something goes wrong, we intuitively understand we're going to have behavioral deficits. And we come across words like inattentiveness, impulsivity, hyperactivity, working memory problems, disorganization, planning, right? But the link between these systems and these behavioral deficits is not clear-cut. We don't always know exactly how that path happens. So it's not black and white yet. It gets even worse when we go back. So the forward model is not entirely solved, but I think what I started calling the reverse inference model is even rougher, right? So when we see inattentiveness, is it this system that's off? And that's not clear, right? And we see this, nowhere else better than, for instance, some of the neurodevelopmental disorders that we deal with, like ADHD, which is very quite prevalent and has high impact due to early onset and problems with adherence to treatment, for instance. And what's interesting here is that, so even though the etiology for a long time, so this is the 90s, includes catecholaminergic dysregulation of frontal cortex, which is part of the attention systems, contributes to attention systems. The overall mechanistic, phenotypic, clinical heterogeneity is very high. And so what does that mean? It means different people show symptoms differently, respond to treatment differently, making treatment design and monitoring difficult. So it's almost like these behavioral deficits are like a canary in a gold mine, right? There's a lot of heterogeneity that we know. And so what I'm going to talk about and sort of the motivation for us, we have two paths. One is kind of trying to speak a little bit to this forward path of finding a way to refine some biomarker, a biomarker of at least some subset of the system that can account for or predict some manifestation of concrete deficits within this bigger sort of family. And then our efforts to sort of think about methods, approaches to resolve this reverse problem. So if you have some symptoms, how do you figure out which source is the source of those deficits? All right, so this story starts here. So I'm starting with that first part of the biomarker part. And so this is many years ago now, really through collaboration with Jim McCracken and Sandra Liu here at UCLA. There was a large effort, a clinical study to look at cognitive control improvement through several different methodological pathway, guanfacine, stimulants, and a combined treatment. And as part of this clinical trial in the sort of pre-medication, the baseline assessment, there were measures collected, EEG-fMRI, other measures, during a number of cognitive control tasks. And one of these tasks included the spatial working memory task. And this was important because working memory, which for instance, in this case, is the idea that we can maintain information in memory for short amounts of time. So in this task, what you do is you encode some spatial information, you maintain it in memory, and then you have to respond whether the probe is in a location previously seen. And so the idea of working memory is really about that maintenance in that time when you don't actually see the dots. So the example we used to give is remembering somebody's phone number. Doesn't happen anymore. So, remembering your PIN so that you can unlock, some account. That's working memory. And of course, there are regions in the brain that actually maintain that information over time. So impairment on this type of task is fairly high, showing medium, large group differences. And so there was a question of what's going on. For us, the question became, well, if there's a problem, or not disorder, but a problem with working memory here and behavior, or is there a problem with attention control? Because there's a lot going on in the task itself. So maintenance is right here, but we're encoding here, we're fixating here, we're making a response here. Any piece of that sort of sequence could impair performance. And that was our original motivation. So to get at the time course of this process, we looked at the EEG data because we have the temporal resolution, of course, and we can look for this sort of epoch, for this event. We can average all the EEG responses. We can extract power in the oscillatory bands and come up with another event-related response to look at the increases and decreases in the power across the window and ask, well, where is the problem starting? And what we found was a very sort of large effect phenomenon, which is that during the encoding period, which are these blue periods for ADHD and typically developing kids at low and high load, we have these decreases in alpha power, 8 to 12 hertz power during encoding period. So this was sort of observed in many other places. That's not new. But what was new was that there's this big, large effect size difference in kids with ADHD. So you can see this is a little blue, less blue up here. And the reason this was interesting to us, other people also found it at the same time. That's what this is all about. So this is Ali Mazaheri working with Ali Jensen. It was happening around the same time. And so for us, the insight was, oh, well, there may be things going on before this maintenance that are contributing to these problems in the behavioral attention, behavioral manifestation of attention. And in fact, maybe what's happening is that the modulations during encoding can actually account for maintenance working memory processes. And that's actually what we found in the original data set. So this is the full time course here. So you see these decreases during encoding and probe. In the middle, you actually see this increase. This is an alpha increase, interpreted as sort of visual cortex blocking. But the important point was that This is also enhanced in ADHD relative to controls, whereas the alpha decreases are also enhanced in the modulation magnitude in the controls, typically developing kids. But when we look at the relationship, what's happening during coding is strongly correlated with what's happening during maintenance, and sort of can explain a lot of the variance in that maintenance process. So this was important for us about thinking about the source process that's contributing to the behavioral deficit. And got us thinking, well, oh, is this a useful biomarker, right? I mean, if it's related with something important in explaining working memory, maybe it can tell us even more about the deficit. So we sort of set out on this sort of journey to figure out what it is. And we came to a couple conclusions. The first thing we did is replicate in a couple different sort of populations. We also, Georgia here did a nice meta-analysis, found that the effect size replicates in other folks' data, so that's good. She also reported in the original, actually, clinical trial, there's a little bit of a normalization. So these changes start looking a little bit more like in the control group after combined treatment. But the fact was that the group effects that we found were just group effects, but you can't really predict diagnosis. So this isn't new. This is sort of common in many mental health conditions. The distributions of these alpha modulations between groups is highly overlapping. There's a lot of heterogeneity. So we don't think about this as a diagnostic biomarker, which shouldn't be surprising in this domain. But what we did find is that it's a very useful, strong functional biomarker of visuospatial processes. And so I'll show you a little bit of data that supports this. Here's a result of a regression, sort of just a chunk of the regression, that shows the relationships between alpha modulation, just during encoding here, and several factors from a factor analysis of neurocognitive assessments. So what we see here is a significant relationship. This is the regression coefficient between alpha modulation and symptoms. So the more decreased, the larger the effect size of the modulation, the lower the symptoms based on SWAN and CBCL scores. It also relates to tests of so-called executive functions like trails, like essentially Stroop task-like switching. And then we have an association with some academic achievement measures, which include reading comprehension, as well as math, and then task performance on this spatial working memory task. But interestingly, we don't have an association with a subset of the academic achievement measures that really focus on reading, basic fluency, basic reading rate, accuracy, spelling. So this is actually a really nice association, kind of by accident. We were looking for a relationship between the neurocognitive outcomes, but this provides contrast to show that this measure is leaning towards visuospatial processes. A couple of our students actually did some path analyses work with this, relating the alpha power to individual symptoms, and they sort of added a little tidbit to the story by showing that this relationship between this and this is in part mediated by the relationship between alpha and these cognitive tasks. So there's really a relationship between the outcome symptoms through the relationship to some of these executive-like but visuospatial processes. Okay, so we have a functional link. And so the natural next question is to look at the neural mechanism and try to narrow down what is this telling us about the brains within these sort of circumstances, within ADHD, in particular within visuospatial processing. Now, at this point, we already know quite a lot about alpha oscillations, right? They're not all that new. They've been around for about more than 100 years. And we know two things. In general, alpha power increases are interpreted as sort of blocked, reduced visual processing when recording from visuospatial cortex. Alpha is not limited to the visual cortex, but that's what we're focusing on. And then decreases are associated with the opposite, which is greater visual processing. I'm loose with my words because there's dispute over what that means, whether it's information flow or interaction, et cetera. But this is a fair representation. So you see this here. When the eyes close, less visual input, alpha power increase. So that's what you see in these little squiggles. Here's a representation in the power spectrum with more power in the eyes closed conditions. In this case, you would see this most strongly over posterior electrodes. And then finally, at the end, you see the alpha modulations, like what we see in the spatial working memory task, moving around the scalp depending on the specific spatial orientation of the eyes in a task. We also know from many, many, many years of work in animals, we have putative generators in occipital cortex, thalamus, from really just a huge wealth of work. as well as some evidence for causal influences when we stimulate or interfere with function in parietal frontal cortices. So we have evidence suggesting that these neural regions or the network associated with these changes should be involving some of these attention control networks that I introduced at the very beginning. So that's what we set out to do. And so this is now a very old study. This was published 2016, where, as Shantanu mentioned, we had the crazy idea of bringing in kids with ADHD to do a concurrent EEG-fMRI study. And so we lost many of those recordings, which led to really actually very interesting selection problem, because what I'm reporting really pertains to the most inattentive symptom children, not so much hyperactive, because motion essentially made the data unusable for a good chunk of children. So it's a very interesting sort of phenomenon we have to deal with in this domain. But we conducted concurrent EEG-fMRI, and the concurrent is important and on purpose because it eliminates between subject variance and allows us to ask questions about the mechanistic associations with these alpha power modulations. So we have a small sample, 30. This is all boys to keep it homogeneous, performing the spatial working memory task. So what do we get? So here's the first finding. What we found is that when these modulations occur during the encoding, we have two things happening. The first thing is that activity in the occipital cortex goes up, which is known, reported previously. But what we really added at that time was that if we look at the connectivity, the functional association of regions in the occipital cortex, we see that it strengthens with a number of regions spread out here in yellow that qualitatively sort of look like maybe some parts of the frontoparietal network, okay? And so that's how we interpreted this, that's, we can, in this task, interpret these modulations to really be associated with this recruitment of this visuospatial network. We also found interesting differences between our groups. Again, very small sample, but it was interesting because it went along with the EEG data. Namely, we see that the relationship of alpha with activity changes is stronger in the TD group. Negative in this case is stronger because alpha goes down, so we're dealing with a negative. But then the opposite things happens for connectivity. There's a stronger connectivity between visual cortex and these other regions in kids with ADHD. And we interpreted this as sort of a compensatory interaction between regions because of sort of insufficient sort of engagement of the visual cortex, or not paying attention, I suppose, not encoding sufficiently. And a part of the reason was that we saw something in that original EEG data where we had elevated alpha power during the maintenance in the ADHD group. So we thought perhaps these are related phenomena, that there's something different happening during maintenance because of what happened during encoding, because encoding didn't trigger certain encoding processes sufficiently. And then one little piece here, this is valuable. So these are correlations of the neural effects. The Y here is the association between alpha and connectivity. So in this case, right occipital superior region connectivity. But what I want to show you is that relates to performance on the task, controlling for age, well, just age in this case. And what I want to highlight is that these dots, these blue and red dots, don't split, right? Not like in the symptoms here. So that means that these relationships with performance are not driven by the group difference. They represent a relationship between this network, functional network, EEG, and performance on the task. So we're putting pieces together for the interpretation of these alpha modulations from sort of various directions here. All right, so that was a sample of 30, which is not so much. So what we got a couple grants, luckily, that allowed us to bring in a whole lot of people. So here's the original sample that Sandy Liu and colleagues collected many years ago. And so we invited as many people as we can to come back and do a second session. So now this is about 10 to 15 years after their original visit that they came back. We did a longitudinal follow-up, including full assessment, EEG, and MRI, which I'll show today. We also collected DTI, and these folks are now adults. Okay. We also have the original spatial working memory task, but a couple of very interesting sort of rest control conditions where people are just opening or closing their eyes. That's the eyes open, eyes closed, or passively viewing the dots, no working memory, no encoding. Okay, so we wanted to see what's going on with these networks and these passive conditions. And this data is under analysis. So next year, I can give you a lot more, but there are some really nice previews. The first is this. So with an N of 160 now, we are replicating the connectivity patterns. So these are connectivity patterns of here, for an example, right superior occipital seed. And these superior regions are the ones we're finding are the most tightly connected with alpha modulations, more so than the ventrolateral regions, during encoding, during probe, and during passive viewing on the dots. These are just a couple of sort of slices, axial slices here, so you don't see a full overlap. We'll look at that in a moment. But what we're seeing is essentially a very similar pattern of functional connectivity between the seed and other regions during encoding. Okay, as the alpha power goes down, stronger modulation, we get stronger connectivity. The patterns are very similar for encoding and probe, but almost absent for the passive condition. And here they are viewing the stimuli. The brain is activating. We're getting alpha. We're just not getting the connectivity, which actually sort of makes sense with what's going on. And there's some very interesting undertones in terms of how we interpret alpha in general. But for our purposes, it's showing a very nice replication of what we got before and suggesting that alpha can be related to this functionality, to this functional connectivity during this process, but not perhaps when you're just passively viewing things, okay? Now we wanted to quantify or, well, qualify these networks a little bit more. So we did an overlap analysis using this dice coefficient by taking these patterns here in the earlier slide and then overlapping them with some known networks to just to give a little bit of interpretation. What are we seeing? Because it's hard to visualize. And so that's what you see here. These are the dice coefficients for a couple different networks. And then here's encoding, probe in the second row and passive in the third row. And right away, you can see some interesting things. The passive condition has no overlap with any networks, which makes sense because there wasn't much connectivity, so there's nothing to overlap. But we also, within the encoding and probe condition, see that we have no overlap with the default mode network and also no overlap with the limbic network, which is, it's always nice to have a sort of reference condition where nothing happens, that is a voice confidence that it's not just, it's overlapping with everything. And then during the actual task, encoding and probe, we're starting to see overlap with the dorsal attention network. This is the primary point of overlap, which is nice. This is what we know. This is what we saw before. But 10 years ago, we saw it with 10 people or 30 people. Now we see it with 160. So that's great. We also see overlap with parts of the frontal parietal network, sometimes called control network, but I took it away because I don't like to call anything control. And then the somatosensory, the visual network. And for those of you studying ventral attention network, there's a very interesting dissociation where the ventral attention network appears to be primarily connected to during encoding, which I think makes some sense in terms of interpretation, but kind of a useful side finding. So this is great news. We're starting to see what we saw before, but way more power to dig a little deeper. We also have these eyes open, eyes closed conditions. And they're interesting because they allow us to say, well, what happens when alpha power goes up during, let's say, eyes closed conditions? So is it the same network, or is the network breaking down now? What do those represent? Is it the opposite of what we see when you get engagement? And here's what we see. This is actually super, super interesting. What happens during eyes closed, so alpha is going now up, which we know means less cortical activation in general. We do indeed, in the bold signal, get a little less activity, but we get stronger connectivity. The network isn't breaking down in connectivity. It's still connected. There's just less activity. So it's almost, if you think about it, that you have a network that has a default property to it. It's there when you're just resting between the visual cortex and these dorsal attention network, but it's sort of like modularizes. It's less active, but it's still connected during the eyes closed when you were getting these bursts. And you see that here. There's a lot less connectivity. And this is just a spatial measure, so it's not everything, but it's showing you that relative to different networks, the greatest overlap we see here is with the dorsal attention network. This somatosensory should be in the visual condition. This is an error. The other place we see the connectivity is the visual network. So essentially, and I think this has actually been published in a smaller paper many years ago by John Fox, that there's this modularization, almost internal coherence within this network when you're sort of just eyes closed, hanging out, and there are these alpha bursts. So that's actually pretty cool to learn. All right, so now the outstanding questions we have are, what is the relationship to symptoms, performance? and the longitudinal effects. And this is, I'll have way more in a year. We're working on this, and this is going to take a while to get through. But we have some tidbits here. The strongest relationship we see so far, the most reliable is what I mean about, in statistically speaking, is during the eyes closed. So this phenomenon of eyes closed, where the activity goes down, but the connectivity goes up, if you think of that as this modularization of this network, actually relates with higher symptoms as well as worse performance during the later task. These aren't overwhelming correlations here, but the range of these symptoms is not high. But they're interesting. They're very interesting. I mean, the performance relationships are a little bit stronger. They suggest that this is actually perhaps representing kind of this disengagement from the visual world during the eyes closed condition, during a rest condition. And the propensity to do that predicts how you're going to do on the task later on. So what state you're in, perhaps, right? Are you sort of kind of already drifting away from the task, and it's really easy for you to get into that disengaged mode during the actual task. Now, the second thing I can say is we're really not getting relationships of any of the connectivity effects during task with the task performance, which is interesting. So why not, right? And we'll see why not. But one thing we're starting to explore is that in other data, we've seen this now in 2 samples. This will be the third one. One of them is published, that in adult samples, we no longer show group effects that we saw in the children in that original study. So here you see nice little blue bursts encoding, probe, and in fact, you don't see bursts in the other direction in maintenance. There's sort of this sort of steady engagement in the visual system, but no group effects. And we've seen this a couple times. This is something we're very actively researching because in the younger kids, here's the developmental trajectory we saw. What we saw was that these alpha decreases, get stronger, keep getting stronger as kids mature from about 7 to 10 to 11 to 14 groups. And originally when we looked at this is 2014, we noted that this kind of looks like a developmental delay in the kids with ADHD. They're going in the same direction. They're just kind of behind. In this broken line condition is a high load condition. So it's a harder condition. It's more pronounced here. They need a little more oomph to get there. So it is certainly possible that in this particular system with maturation, we're getting to a point where we can't tell a difference. Now, the question is now super interesting because it opens the question of, well, what other system is not working right because we still have behavioral problems, or are there in adults different subgroups that follow different trajectories and the average washes out? Okay, so that's what we're looking into. We just don't know the answer, but it's sort of the next question. So here's my interim summary, so it's good time. We're feeling pretty good about this interpretation, at least for that modulation during this type of a task. The degree to which you modulate these oscillations is relatively tightly in our data coupled to the recruitment of visuospatial attention circuits. Now, this is what I just said. We're really interested in understanding the developmental trajectory because I think it'll be just a wealth of data, how these change over the course of maturation, and might tell us a little bit more about how working memory happens and how the initial encoding relates to that. And we're exploring this idea of, well, can these rest conditions tell us something about how prone you are to this engagement, and can that be predictive about behavior? Can it be behaviorally important? And the reason that's interesting is because when you're thinking about something you want eventually to be applied, assessment with an eyes open condition is a lot easier than assessment with a complicated task. And so it's just, from a practical perspective, it has more value for scaling to a clinical application. All right, and then forthcoming, just to let you know, in about a year's time, maybe a year and a half, we're integrating the EEG and fMRI concurrent data with anatomical correlates. So we have DTI data. If anybody wants to do multi-data fusion, call me. We have a lot of data that needs to be analyzed. And we also have a really amazing collaboration with Karen Mollinger in the UK. She's a physicist with unique skills, probably one of the only in the world, to do laminar EEG, fMRI at 70. And so she's working with us to understand whether these alpha modulations relate to layer 1, layer 2, layer 3, layer 4, which will inform which way is the information being processed. What is the likely directionality of information flow? Okay, so it'll help with the interpretation of some of these effects. All right, so that's a lot of good stuff coming. Now, so that's where we got to. We think we have a biomarker of visuospatial engagement. We feel pretty good about it. And so the question now becomes, all right, well, that's nice. Is that going to inform, if a parent comes up to me and says, well, what do I do for my child? Based on your knowledge, you know, I can say, well, you can measure some alpha and, you know, look at it, but I'm not going to give you anything useful, right? And that's the reverse problem. When I see when I hear or see an attention and behavior, what can I do to use that information to offer useful information about remediation or source of the problem? And so in ADHD, that primarily a wonderful context to study this is academic achievement. And we're talking here about translation, right? So let me illustrate the problem a little bit more. I've shown you lots of these pretty pictures of the alpha going down and correlations, and they're all significant, but you see how scattered they are, and they're small. And this is very common in the between subjects sort of analyses. And the point is that if a child scores a low score on reading, now, statistically, this would predict weaker modulation of alpha and therefore a weaker engagement of the score text. But I mean, you know, there are kids here who score low who have high and some who score, right? It's a pretty scattered correlation here. And this is pretty common in the relationship between actually medication, cognitive training, cognitive testing, and the relationship with academic outcomes. So the second piece is really motivating us with this desire to improve translation. What's going on? Well, we have a pretty good idea of sources of variance, right? We have a lot of sources of variance when we go and measure behavior, these inattentive symptoms in natural environment. We have system interactions. We know visuospatial attention doesn't work alone. It interacts with arousal, it interacts with motivation. It also varies with context. So how these kids do will vary from the lab to home to school. We all know this. We sort of ignore it in order to isolate systems in the lab, right? And then of course, we have diversity. People come with sort of different histories, different baggage, different impacts on their entire system. And so this was what we started going after. We said, well, we got to deal with these sources of variance somehow. And what we decided to do is start with context, purely because it's the most accessible, lowest hanging fruit. It means we can take a marker we already know and start moving into different contexts, this tiny little step to start understanding how context affects these processes and how it contributes to the behavioral deficits. So here's a study we did with my collaborator. Here's Jenny Grammer, also Sandy Liu. And we decided to run a study where we're going to use our EEG signals to try to identify with the goal of ultimately optimizing learning environments for kids with ADHD. So context here are these learning environments. And the idea was really straightforward. It's the simplest thing we could think of. We can vary, we can create a little neuroscience class for kids. These are going to be small kids, elementary school. We're going to vary the contextual factors by varying the amount of teacher support and interaction. And this study started right before the pandemic. So this was not supposed to be virtuality, but it became virtuality because that's what we had to do. But the idea is very intuitive. So we have a couple of conditions where we vary teacher support. The kids can work on they're building a neuron pipe, a pipe cleaner neuron individually with the teacher helping them. So they're doing a hands-on activity alone. Then they have an in-person back and forth, some questions, sort of discourse. Then we have a Zoom condition where they're doing the exact same thing, again, with an individual. So there's interaction, but we're moving into online context. And then we have a neuroscience video. So there's no interaction with the human at all. And the idea was, what we expected was that the in-person condition would probably be the most supportive, especially for kids with ADHD, and we should see greater engagement. All right, so here are kids. We have mobile EEG headsets here. So the important thing is that the kids can move around. You can see she's holding her leg. They can wiggle. That's not a problem. We sort of deal with it post hoc. We recruited 80 kids. We have about 60 in the analysis. And we had about 10 minutes per condition. And of course, what we recorded was alpha power, since that's what we know now. We have an interpretation. So let's start with what we know. And just as a reminder, more visual attention is less alpha. So there's always a negative correlation in our interpretation. And we're not looking right now at, as you'll see, at these modulations. We're looking at alpha power during our 4 activities. So what do we get? Well, here's the first result. So this is a scalp representation of the alpha power. Here on the left, we have power from the occipital electrodes, on the right from the central electrodes, because they're a little bit different, so we kept both. Different bars are different groups. And the first point is we don't have group effects at all here. And 60 is not a big sample. We tried splitting by diagnosis or symptom stratification, and the group effects are really not very big. There are some, but kind of weak. But there are effects in condition. And so at the very least, that's saying that the condition effects are larger than the group effects, which perhaps isn't all that surprising. Within subject effects can often be bigger than group effects. Now, the second thing is we have some validation conditions. So eyes closed, eyes open. We know there isn't much going on. We have lots of alpha here, right? That's expected. That's what we know should happen. It's just a sanity check condition. Alpha power goes down, visual engagement goes up when you open your eyes, and then even more so once you're actively engaged in some kind of an activity. So this is good. It's a good validation. And then we have another drop, which is significant in the central but not occipital electrodes, from the video condition to the interaction. There's no difference in whether you're interacting with your teacher on Zoom or in person, at least for 10 minutes. And then there's a further bigger drop when you're working hands-on building your pipe cleaner neuron. So we see an effective context. We get some proof of concept that you can identify conditions in which engagement is the strongest. In this case, it's the individual work when they're building, and identify conditions where the symptoms might manifest a little bit more, where the engagement is weaker. For those of you interested, I haven't introduced this, but the effects in the slope of the entire distribution of the power spectrum are actually quite similar. So here's our alpha, and we can always look at the slope of this distribution to see what is essentially the distribution of other oscillations, high frequency, low frequency oscillations. And the slope is interpreted in terms of sort of state of the brain. It's interpreted in different ways. Some folks suggest that the flattening of the slope suggests more noise in the system. Some folks suggest, as the movement from eyes closed and eyes open to task, that the flattening of the slope is something that happens naturally when you engage in visual processing. There's more interaction. There's less low frequency, sort of more high frequency power. And the only point here is that the direction is exactly as with the power. So the steepest slows are present for the eyes closed, eyes open in the video condition. And then this flattens with the flattest slopes for the highly engaged condition based on the brain data. But now it's based on the brain data. So we should say, okay, well, does this predict anything behavioral? So far, you're just showing me some brain signals, but we don't know how to interpret it. Now, we video coded the behavior of these individuals minute by minute for the entire duration, using a series of categories used in various school studies and ADHD school studies, and use that as a gauge of our sort of attentiveness from a teacher's perspective, from a behavior perspective. So then we can look, and I'll focus on active engagement here, which is, if you see here, answering teachers' questions, following a teacher directions, doing what they're being asked, which is kind of like the attention control definition. And then we can look at the attentional engagement, and I broke this up into 4 quarters for each task so that we can use time on task as to introduce a little more within subject variability of how these change. So you see some nice things. The first thing you see is that the behavioral ratings of active engagement, the video condition, are at floor. You're just sitting passively. But then in the in-person interactions, you actually get a decrease over time. So the different colors are different groups. The significance is for the mean across groups, of course, because we have a small sample. And overall, in these 2 conditions, engagement decreases. So no surprise, in 10 minutes, these kids get bored of interacting with the experiment on some boring neuroscience conversation. In the individual condition where they're building a neuron, it actually goes up. And so what actually happens at the beginning, they're kind of not really interested. They don't know what they're supposed to be doing. Then they get their hands on this neuron. And they're like, oh yeah, this is cool. They're really, really sort of captivated. Good. And now we can see, well, does alpha do the same thing? Does it track any kind of this behavioral phenomenon? And the answer is yes, it does. So the alpha power is flat in the video condition as well. it increases with time in the Zoom and in-person condition and decreases with time in the individual condition. So it's tracking what we're seeing in the behavior. Okay, and this is really cool because it's telling us that we can track this visual engagement in natural environments to understand contexts that facilitate or hinder individual child sort of engagement, which again is the starting point to understanding what are the sources that are driving problems for them. We have a couple of papers on this, but this particular data we're writing up right now. And then we can move, okay, so this is where it starts getting complicated. This is the natural environment, but it's interesting. So the first question is, what about event-related alpha, the lab task that I've just been showing for 40 minutes? And we measured it in these kids as well. It's a lot messier, as you can see. We only have a very small sample in the ADHD, but the key effects are there. There are these decreases during the encoding and the probe. You see them here at the back of the head as well. Okay, and we have these large increases over the central electrodes, which we're sort of ignoring for the moment. We need to dig into them a little bit more. But the key point is this: when we correlate the alpha decreases in the lab condition, so these are decreases relative to baseline during this task, with the alpha power changes during the activities, we get a beautiful flat line. And then we've also tried to take pairwise differences in this alpha power conditions and activity to kind of approximate it something like an event-related change, like relative to a reference. And that's also a beautiful flat line. So what's that saying is that these modulations are not capturing the same variance that we capture in the alpha during the activity conditions that relates to the behavioral symptoms, the kind of behavior that relates to symptoms. Okay, so we're splicing the variance here. But if we correlate the same activity during alpha, during the class activities with the background activity during this task, so not event-related modulations, but the background activity, we get it back. Okay, so that's saying that the background alpha power is capturing different sources of variance, different interactions perhaps, than what we measure in these extremely finely controlled modulations in the lab task. So that means the lab task is not going to tell us about active engagement. It'll tell us about that visual process, but not the symptoms which the clinician and teacher observes. And you know why? Well, I started hinting at it in terms of system interactions and diversity. So let me show you, we can see that in our data a little bit, how that plays out. And we have to discuss them together, and you'll see why. Here is just for demo, one of the trends, right? So alpha is increasing, and just to remind, more alpha, less engagement. So less alpha, more engagement, vice versa. But if you look close, so you see in certain places there are differences between the groups. And in this case, the ADHD group has lower overall power, even though they have the same trend. And this creates this wonderful substance paradox in interpretation. So less alpha, more active engagement within individual, but less alpha, more ADHD between individual. What's going on, right? This is classic Simpson's paradox. Now what, right? Does that mean that between subjects, individuals with ADHD have more engaged? That makes no sense, right? And indeed, when we look at the correlation between subjects, it's not there. So what is driving these kind of relationships? Well, we have other measures, and we see that we look at fidgeting between individual, kids who are more fidgety also show less alpha. So it could be that the within subject change in these measures are indeed driven by some degree of visual spatial engagement in the task. But between subjects, there are these other big sources that we are not accounting for. And that's now getting to the system interaction and diversity, because it's between subjects, and system interactions because fidgeting could be motor agitation, could be arousal, could be something else. And it could be driving down those alpha measures overall, that baseline level of cortical engagement, whether it's from the motor cortex or somewhere else. All right, so that brings us to where we kind of are mentally right now. When we're thinking about visual spatial processing and visual control, we're measuring these squiggles, alpha, and we have to remember, we're trying to remember right now that this is, yes, it's representing, in this case, maybe a dorsal stream, ventral stream interactions, but it's also representing all this other stuff. There is behavioral relevance, there's arousal, not even drawn here. And And what that translates is, of course, other systems too, which we're focusing our motivation and arousal, because of course, in the natural setting where we observe attention deficits, we have social interactions, teacher report, success. Do you want to be there? Are you working with kids that you like in your group? Or are you stressed out because somebody looked at you the wrong way? And then on the arousal side, fatigue, obviously, right? Social anxiety. And these are constantly moderating, interacting with whatever your visual system and orientation is doing, and the behavior that the teacher is observing. And this is a little chart that we put together for Grant to explain this sort of to work through how this can go now wrong, the kind of confusions we can arrive at. And so, here's behavior. This is what the teacher sees. Let's now pretend we can actually measure visual attention. We'll take her alpha, we'll assess, we're gonna have a gauge of one system. And so in an ideal setting, we have everything is well, behavior is good, visual attention is good. In the top half, we see we can have a situation where the visual attention might not detect a problem. The system is activated, but the behavior is still not great, and we would not be able to find a solution without looking at, let's say, arousal. This is all made up, but just making hypothetical scenarios. And then we could imagine scenarios where we have a different problem, where you have the behavior is poor, the child is inattentive, the teacher takes their handy dandy measure device, shows alpha is high, and says, well, their visual attention system is disengaged. Let's intervene with some kind of a program, and the program works for one of the kids, but not the other because of missing this additional info. So it's important to think through at how these interactions can lead to very real confusions in behavior and therefore obviously problems with remediation. All right, so let me wrap this up. So where are we? We started with looking at the sort of forward problem of finding a biomarker for some kind of a system within this bigger system of attention control. And I think we're feeling pretty good that we can interpret what's happening with at least alpha modulations during the actual task, right? We're still kind of learning about how to interpret it in natural settings. We are very, very interested and working hard on the developmental elements, because I think those will be a gold mine of information about how this process changes. And of course, integrating, working on some multimodal fusion with anatomical correlates, asking whether there's a particular system, thalamic pathway, direct longitudinal pathway, that's anatomically contributing to these differences in visual spatial processing, or if it's a purely functional problem. So more likely that it's associated with those neurochemistry differences in the brain. But now where we're sort of, our future here is trying to make sense of this, making sense of this reverse problem. And what I hope I showed you is that there's some value in going into the wild and dealing with this variance in a somewhat controlled fashion. So we started with the easiest one we could think of, which is context. But of course, we can't run away. Once you go here, you sort of can't run away from diversity and sister interactions. And what we're doing is running away a little bit from diversity and hoping to focus on system interactions for a bit to make the problem different. And so where we're working towards an individual approach. This is work with Dr. Emma Urton, who's engineering at Ohio State, to really build in with these mobile systems additional measures so that we can directly measure these arousal systems and these motivation systems and integrate the data, ask the question explicitly for this child, which system is driving the neural measure, which system is driving the behavioral deficits, and see what we can find out and whether we can feed that back into our neuroscience to understand how these systems work. And then one last piece, we're working on an analytical project. This is Sarah Karalunas and Vince Calhoun on extracting more refined measures to complement the oscillations. I've sort of been living in an oscillations world, but it's not the only measure. I hinted, there's the aperiodic slope. There are other measures that can describe the system of the brain. And so that's what we're trying to do at the same time. And I believe, and it isn't unless we tackle these problems that we can actually answer some of the most important questions in the clinical domain, and not just ADHD, but these are questions which sort of cover in regular conversations in neurodevelopmental disorders. So we're hoping to get a little closer to answering them. And it's always too many people to answer, to think. but they're all here. Support from NIH, Brain Behavior, Klingenstein, and many, many collaborators. All right, thank you. I think I'm great on time.
It's a great talk. From my reading, I think there should be 3 talks that we should be giving. And so focused on alpha waves and attention by itself, because that's kind of, for those who don't know EEG or were not in the field, a bit hard to follow. And then the context and naturalistic paradigms, which I think is very complex.
It's very
I
complex.
know we looked at a subset of those things, but so that's I think the most interesting part. And the translational aspect of it, which you did touch upon.
Yeah.
We want to know more, for example.
Yes, yes.
So any questions from the audience first? I'll just monitor the Zoom.
Yeah, I don't know how the Zoom was coming. Yeah. I think they were actually all the same, but it's a negative correlation. So the question here is about, let's start with the practical one. This, right? So they're actually all going in the same way. This last one, the overall in the individual condition, what's happening is that the alpha power is going down with time. In the bottom one, this top, it's kind of flat here, right? So you have this, I don't know what the fit would be. I don't know if this is a cubic or what, but you have a big jump at the beginning and then kind of a flattening. So this isn't well, I actually don't know statistically if these 3 points are a downward trend or not. So we need more data is the answer. I'm claiming that this one is going together with the one above, but we'll need a little more data to show that that's in fact the case. Yes. Yeah, so the question is, why do we need EEG and fMRI at the same time? And the answer is we don't. If you don't want to know about 2 processes in terms of their temporal concurrence. But the benefit is that I can eliminate between subject variance. It's a within subject test of, does the time course of my EEG signal correspond to my bold signal in other process? The instant I do that across individuals and say, is there correlation between EEG and fMRI across individuals? I'm throwing in that between subject variance, which is maybe reflecting the mechanism if there are no other sources, but if there are other sources of variance, it's mucking it up a little bit. It's messing up the variance. So it is important. They're not the same signals. I always say, EEG, fMRI are not the same signals, and we weren't assuming they were the same signals. The reason in this case it makes sense is because we know alpha oscillations would be associated with a distributed network that may involve thalamus. And we knew that we cannot make big conclusions about thalamus from EEG alone. And so in order to get a full view of the brain with subcortical contributions, we need the fMRI. And so that's actually what pushed us to go concurrent. And we're looking currently at connectivity for the thalamus to complement the analysis of the visual cortex connectivity to answer that question specifically. Yeah. Does it change their behavior? So the question is, does putting the net on the kids in the scanner change sort of their behavior? It's a great question. I'm thinking whether I have a quantitative answer to it. Okay. We also collect, I can't tell fMRI. We always collect data in the scanner and outside the scanner. So I can tell you that the correlations of that data is significant. So we think that we're capturing the same thing we would capture outside the scanner. So maybe I stop there and not dig a bigger hole. The fMRI, I don't know because I didn't collect non-EEG data from these particular individuals. Traditionally, with this methodology, the damage is bigger for the fMRI side, for the EEG side than the fMRI side. And we try to keep the testing very short on the MRI side to sort of deal with that problem a little bit. So try to collect data early on when the kids are still pretty still, leave our anatomical collection till the end, take off the cap halfway through. But I can't give you a more quantitative answer than that. Oh, I think there's some questions here online too.
Mostly about
Watching it. Okay. All right. Okay. Oh, yeah.
and some
Yes.
asking research papers and materials.
Oh, please contact me. Yes, yes, yes. Please contact me. I'm happy to share slides or materials or whatnot. Yeah. Yes, Leo. Yeah. Yes. Yes. So the question is about investigating alpha movement. We have not done it, but it is being done, especially in the mobile EEG domain, where a lot of the questions being asked, like actually Nancy Asutana's lab, right, who was here for a long time, is looking at moving around, right? So I do know that the work is being done. Movement does affect both ERPs and alpha. It will modulate the signals in different directions depending on whether it's purposeful movement or whatever it is, and we can measure it and we can track it. It's something we're going to keep looking at. I do think there are two sources that are contributing to our alpha measures. I think that central source is a different source. We're looking into sources now. We think it's very likely related to mu or a motor kinase response. So yeah, TBD. My hunch is that, yeah, it's 100% movement. And something I like to test is whether that movement isn't accounting for some of these between subject effects that have been found before, with alpha power being lower in kids with ADHD. And wouldn't it be interesting if that measure reflected the fact that the kids are more fidgety? Be kind of an interesting phenomenon. And how does that affect the rest of the measures? If you have a system that's active because you're moving you're engaging your motor system. So I don't have an answer. I agree. I would say it's a really important dimension which we're being forced to investigate. And that's kind of what happens with these naturalistic studies, is that you immediately get forced to try to understand things you really don't understand or perhaps don't have expertise in because it's there and we can't ignore it. Great question.
Did you have eye gaze data? Eye gaze?
We don't have it here. We are collecting it right now. Yes, it is. I don't have the slides. So one of the things, we have eye gaze for these kids, the naturalistic study, from video recordings. Okay. So and what we're finding is it tracks alpha really nicely. So I think it is important. It is an orienting signal. What we're doing now in the follow-up is using glasses, which have an eye tracker in them and can do gaze, and use them they're used in mobile studies regularly. And so we're quantifying, because what we found is it's likely going to be a really great tracker, maybe a way better tracker than alpha, frankly, because it's a higher signal. But the video coded gaze is just not as reliable. So it's coming. Yeah, it's coming to see whether it's a good alternate signal or a complementary signal.
I have a lot of questions, but a few of them I'll ask offline. One of them, stimulants and medications. So you did mention those two words. If
yes.
You want to touch more upon how this could be not applied, but how this could lead the way
to testing.
Yeah, testing.
That's a tough question.
Stimulation, not as a means for treatment, perhaps, but also as a probe for alpha activity. Without treatment?
Yes. It's a great question. So how can we sort of weave in what we're doing with medication pharmacological based treatments and this kind of approach to testing? I suppose we can now have a broader range of effects that medication may or may not have. Because if we measure these systems in natural settings, perhaps we can expand our monitoring and analysis of where the medications are helping versus just self-report, for instance. One caveat to this entire sort of phenomenon is what I wish we had is an ability to measure the neurochemistry of the brain in real time all the time. And then perhaps we wouldn't have to be dealing with some of this variant. Maybe we still would. But it's kind of the missing piece. And so that's what puts like a little bit of I hesitate because I don't know if we could observe it, but in principle, maybe we could learn about contexts that are best suited to particular medications.
Because there are MRS studies.
Yes.
Colleagues, you know them, which we are also.
Yes, and I'd love to do more of that. But I'm waiting for some of those techniques that are tracking, dopamine in real time in intracortical studies to be maybe a little more non-invasive. Because I think that's where the money is for some of these questions.
Marco.
Another question, study outside, you mentioned fidgeting a lot.
Mm-hmm.
And in the whole products about one is about the task. One is that it's because we have to about sense itself.
Mm-hmm. Okay.
It's the data on sense itself in ADHD.
That is a great question. I think so. The question is, there's a lot of discussion about fidgeting. There's a framework for interpreting fidgeting in terms of need to reaffirm the self. And so is there any information about that in the ADHD literature? I have no idea. I'll look that up. I'm wondering if there's anybody online who's still from the clinical domain who can actually answer this, because I actually don't know. It's a great question. Do you know?
Yeah.
Yeah, let's do it. Let's do it. I mean, it's really a great question. I know there is a little bit of research into fidgeting. I don't know how systematic within a particular domain that is, or if it's sort of a bunch of dispersed, kind of scattered studies. I just don't really know, but it's a great question. I mean, because then you're asking a question about how do you operationalize self-affirmation? Or is that a reflection of something else? Like, what does that look like? And not just operational, how do you define mechanistically? What's that correspond to? I mean, is that another dopamine signal, or is it something else, or right? Sort of a really interesting question. Yeah, I like that. I don't know.
Well, were any of these kids medicated during the study, or they were washed out? Or?
In all the recordings are washed out. So the original study was baseline, so pre-medication, and all of the studies require this continuing medication for the. We do have that one paper from Georgia on medication effects. They're not as clear, they're small effects in terms of the effects. Yeah.
Great. So this was a great talk.
Thank you. Thank you for having me. I really appreciate it. And yes, please contact me if anybody needs slides or didn't see anything online.
See you next year.